So, stuff you already know, but I feel like I have to say out loud. These are atria and these are ventricles. Okay, so now that we're past that, if you want to see the internal structures of the chambers, you are, of course, going to have to do a section. Logically, what kind of section would show you all four chambers simultaneously? Yeah, coronal section. Now, if you wanted to see a real difference between the sizes of the thicknesses of the chambers, oh, I forgot to tell you something. You could do a transverse section, and that would show you how much thicker the wall of the left ventricle is than the right ventricle, but it wouldn't show you all four chambers, and we'll talk about why the left ventricle is thicker, and most of you guys probably already know. Okay, I forgot to tell you something. If you were coming to my office to look over tests today, don't, because I forgot them all on the other campus. Whoopsie daisy. So they're over there. Sorry. They're very well organized over there, though. Okay, so the separation between the atria in a healthy adult is complete. Okay, so this separation between the two atria is logically called the interatrial septum. Thank you for making words logical. Okay, so that's called the interatrial septum. In the wall of the interatrial septum in a healthy adult, you will have a depression. Usually you can see it best when you approach it from the right side, but you could see it from the other side, but the left one is harder to get to because it's smashed back behind the blood vessels. This depression right there is a remnant of the fetal structure. When you were a fetus, maybe this should make sense to you. When you were a fetus, you didn't need to deliver different types of blood to your heart and your other organs, right? Sorry, not heart, your lungs and your other organs, right? Your lungs didn't need different blood. Does that make sense to you? Because they weren't really functioning uniquely. They needed blood, of course, because they need to do cellular respiration and growth and all that business, but they didn't need different blood. So what happens during fetal circulation is we remove the sort of separation, and instead of allowing the heart to be two side-by-side pumps that are independent of one another, we allow them to have crossover of blood. And one of the ways that we do that is that there is a hole between the left and the right atria. When it's open, it's called the foramen ovale. And after it closes, it leaves a depression, which you can usually still find in a specimen, and that's called the fossa ovales. And if that doesn't close, they usually describe it as someone having a hole in their heart. Does that make sense? So you can find that in the wall of the interatrial septum. You can also find this. We'll talk about later. Okay, so what's true of both atria is that they are thin-walled. They're mainly receiving chambers. I'm not saying that they don't do any pumping, but their pumping is less important than you think it is. And then on the wall of both atria, there is some lumpy musculature, and the lumpy musculature on the wall of an atrium is called pectinate muscles. Now, on the left side, you can't really find it unless you open up the auricle. So let me show you what the auricles are real quick. Look at this. Do you see these two things that look, auricle means ears, that look like a Labrador retriever ear? See it right there? Okay. Those are called auricles. They're this flappy part that if you've got a lot of venous return, they can open up and provide a little bit more space for blood to actually return in. That's the left and the right auricle. You've got them on both sides. Okay, so when we are moving through, we are going to do atria, and then we've got ventricles just generally. So we'll get into the specifics in just a second. So when we move into the ventricles, we are going to move through these two valves. We'll talk about the functionality of the valves in detail today. These are collectively called the atrioventricular valves, which just makes sense, right? Between the atria and the ventricles. Talk about their function in detail. And then you move into the ventricles. What is true of both ventricles is that there is lumpy musculature on the walls of those. And the lumpy musculature on the wall of a ventricle is called the trabeculae carnii, which is impossible to spell. It's got vowels all over the damn place. So you're going to have to study that one, right? Trabeculae carnii. And then in addition, you have these muscles right here, these muscles that project up from the trabeculae carnii, and somebody thought they looked like a nipple, go figure. So these are your heart nipples, your papillary muscles. So we're going to have heart nipples and kidney nipples and apparently brain boobs, all kinds of stuff. So, what?
The more the merrier.
Yeah. So this one has an odd number of nipples. I don't know how to explain that. So these are your papillary muscles. The papillary muscles look like they are going to pull the valves open, but that's not what they do. We'll talk about what they do in a minute. You also have this structure right here, which attaches the papillary muscles to the flaps of the valves. These are called the chordae tendineae, and the chordae tendineae are your heartstrings. What they are going to do is stabilize those valves. There's a cool picture of your heartstrings right there. Yeah, this one's cool. Stick a camera in there. So those are your heartstrings. We'll talk about the functionality in just a second. So how do you, before we get into the deep functionality, tell when you're in the left or the right heart? Well, interatrial septum, interventricular septum. Yeah? And the interventricular septum is far less likely to have a hole in it, although every once in a while it does, because it didn't really start with a hole on all of us. So this is the interventricular septum, and it's pretty muscular right there, and it separates the left and the right heart. Something conceptually interesting that I was confused about for a long time, and I finally did some literature review on it, is I was like, why the hell is the inner surface of the heart lumpy? It's lumpy, but it's not pointy. So it's not pointy like it'd catch a platelet, but it's like hills. And apparently what that does is when the walls of either the atria or the ventricles contract, if they're nice and smooth, what happens is they will adhere to one another like two glass slides.
Oh, it's like a centogonite.
Yeah, exactly. So it's like allowing them so that you can contract fully, but then you can pull apart readily. So that lumpy musculature apparently has that functionality to keep them from adhering to one another when you contract maximally, which I thought was pretty damn cool. Finally thought about asking that question. You're like, why didn't I think of that before? But I hadn't thought about it. So that's what that is apparently, the functionality of that. And they did some simulations in which they make heart structures in which they don't have the musculature, and and then they can't pull them apart because of hydrogen bonding. They just get so stuck because of hydrogen. I thought that was neat. Okay, so the interventricular septum. So that is when you have a coronal section, right? You can tell left heart versus right heart really easily when you have a coronal section, but how can you tell on an intact heart? When you're looking at an intact heart, what you use is landmarks. This depression right here is called the anterior interventricular sulcus, and it will have fat and blood vessels in it, to be named later. And the anterior interventricular sulcus sits right on the interventricular septum. That means that everything on this side is right heart, and everything on that side is left heart. Good? How can you tell when you're in atria versus ventricles? Well, open your eyeballs. But in addition, there is also a depression that runs all the way around the heart, and this is called the coronary sulcus or the atrioventricular sulcus. Runs all the way around, and that tells you atria versus ventricles. If you're looking at the back side of the heart, how do you tell whether you're looking at left or right heart? If you're looking at the back side of the heart, you can tell because there is another depression. This depression right here has fat and blood vessels, to be named later in it. This is the posterior interventricular sulcus, okay? And everything that side is left heart, and everything that side is right heart. Good? So those are your landmarks that you use to figure out where you are. Okay, so now let's start looking at the specific chambers. And you can learn the chambers any way you want. I'm going to learn it in order of blood flow. So we are going to arbitrarily start here. I mean, you start anywhere in the story, right? But I'm going to start in the right atrium. So before we go any further, right heart versus left heart. Which one is going to be associated with primarily low oxygen or deoxygenated blood? Right heart, exactly. So if you were looking at a colorized picture, a colorized model, then all of the big vessels that are blue will be associated with the right heart. Yeah? And all of the big vessels that are red will be associated with the left heart. But does the specimen actually look like that? No, it does not. Okay? First off, even though I cannot convince my 10-year-old, you don't have blue blood in your veins. He literally will not believe me. I don't know how the hell to convince the kid. And he said, this last time I told him this, he was like, I'm going to go ask Dad. I was like, your dad knows computers and music and philosophy. Don't ask Dad about anatomy. I've got my purview and he's got his, right? You ask me about literature and science. You ask him about the rest of the crap. And then he went and asked my husband, and my husband's like, literally no idea. Don't know what you're talking. Pretty sure it's red. My husband still has never seen a cadaver. Like, we have been together for ages, and he has never seen one. And he's like, I'm never doing that. I'm never, ever doing that. The vessels that you see that are blue, the big ones, are going to be associated with the right heart, and the vessels that you see that are red are going to be associated with the left heart. So arbitrarily, we will start right here. So let's get blood into the right atrium. Okay, so conceptually, where is the blood coming from that ends up in the right atrium? The body, right? Systemic circulation. And it is going to come from above the heart and below the heart. Yes? And it's going to come from above the heart through this vessel right here. And this is called the superior vena cava. Which direction should blood be flowing through that vessel? Down, exactly. And it's also going to come from this vessel right here, which is the inferior vena cava, which is going to be delivering blood back from systemic organs below the heart. But, of course, the heart is a systemic organ. Does it make sense to you that the heart should be a systemic organ? So I'm also going to have to be delivering blood back from the heart itself. So this hole that you were seeing right here is delivering venous blood back from the heart muscle itself back into the right atrium. And that vessel will be one that you haven't learned yet. It's called the coronary sinus. It's right back here on the back side. So if you are in the right atrium, you should be able to find your way pretty easily because the right atrium has an enormous vessel on top and an enormous vessel on the bottom. Unless you have an amazing specimen, you probably won't see that. It's there conceptually, though. It has to be there, right? The fossa ovalis you can usually see if you like dig around in there. So starting on Friday when you guys start this first lab tomorrow, make sure that you prove to yourself how things are connected with the heart because you will have either a sheep heart or a pig heart in addition to a model. Stick your fingers in there, push on one side of the valve, see whether it opens or closes, show where your finger actually ends up. Because once you get into here, if you push on this valve from the top side, it works like a saloon door, right? If you push on it from the top side, it will open. Okay? So this valve right here that you are going to run into when you're on the right side is called the tricuspid valve because it has 3 big flaps. It's not only 3, but 3 big ones. Or the right atrioventricular valve. Okay, right AV is okay and it's always safe. So if you can't remember tricuspid versus bicuspid, say right atrioventricular valve. Now, tricuspid valve has 3 flaps. The one on the other side that we'll talk about has 2 big flaps. This one's bicuspid. The way that I've always remembered is try to do right and then buy what's left. Okay, so now we are moving through the tricuspid valve into the right ventricle. The right ventricle is thinner walled than the left ventricle. I think you guys told me last time, but verbalize it again. Why is it functionally logical that the left ventricle is going to have way thicker musculature than the right ventricle? Go ahead.
It has to get blood to all the organs.
Yeah, so basically I've got a lot of tubes that I have to deliver blood through, and some of them are working against gravity and some of them are all the way down at my toes, right? And so I have to generate way more pressure to get fluid through a 16-foot hose than a 6-inch hose, right? So you need to generate more pressure. The left ventricle is going to be way thicker. So the right ventricle is way thinner because it's just delivering blood to the lungs. Okay, so when this ventricle contracts, what it's going to do is it's going to push on the back of another saloon door, okay? And that one, notice the direction, right? So if you push on the back of that one or the bottom of that one, what it's going to do is you're going to push it open. That is a semilunar valve. It's called the pulmonary semilunar valve. And the pulmonary semilunar valve is going to deliver blood into this big artery called the pulmonary trunk. And of course the blood is headed through the pulmonary trunk to where? The lungs, exactly. The pulmonary trunk is actually an artery, but it's not usually called artery until it splits into the left and right pulmonary arteries, right? So note to self, you have just proven to yourself that not all arteries are oxygenated because this one is pretty damn important and it's deoxygenated. Okay, so what I'm going to do is I'm going to deliver blood through the pulmonary trunk and then the pulmonary arteries. I'm going to deliver it to the left lung and the right lung. It's going to go through the pulmonary capillaries. It's going to change gas content. And after it changes gas content from the gas exchange in the lungs, then I need to deliver it back to the heart. To what chamber? Left atrium, exactly. And I deliver it to the left atrium from here. The left atrium looks really small. It's not smaller. The atria and the ventricles on either side have approximately equal volumes. This one's kind of smashed back behind, and remember it's twisted too, right? Okay, so the blood that comes back to the left atrium comes through two pairs of vessels, the right and left pulmonary veins. These are veins, even though they're oxygenated, because they're delivering blood back toward the heart. So the right and left pulmonary veins, which direction is blood flowing through those? In, right? So do you see that if you know what to expect in the atria, it's really easy to tell the left atrium from the right atrium. The right atrium has two big holes on top of each other, and this one has four small ones lateral to one another. So this is the right and left pulmonary veins. It's going to go into the left atrium through those guys. They're most easily visible from the back. Okay, so now I am, of course, in the left atrium. From the left atrium, I look at this saloon door, and if I had pressure buildup on the top of that one, I would push it open, and we'll talk about valve function in a second. This is the bicuspid, or left atrioventricular valve, or there's a very common clinical name for it that has to do with its history. It is very commonly referred to as the mitral valve, clinically. Okay, so mitral valve, let me tell you why it's called the mitral valve. It just reveals a little bit of the history of anatomical study. So way back when in Western civilization, if you were going to be an anatomist, you were either Catholic or you were burned at the damn stake. Those were your choices, right? So not surprisingly, there are lots and lots of religious metaphors and anatomical terms. This is one of them. That is called the mitral valve because the big boss and all of the underbosses wore these cute little hats called mitres. And like the Pope wore one. So have you ever seen the Pope wear this hat? Yeah. Do you see it? The papal mitre? You see it? This is just like an upside down. That makes sense? So that is the mitral valve because somebody said it looks like a papal mitre or a bishop's mitre. Does that make sense? That's the history of why that's called that. Okay? So the mitral valve, and it's really commonly referred to as the mitral valve. Interestingly, anatomists, like the academics and educators, have different terminology than the clinicians. It seems like we would get it together at some point, but we haven't. So there's times in which we're just arguing, and we insist that this is the best way to do it, and they insist that they're not going to change. Okay, so this is the mitral valve. This one has two big flaps, and this one has three big flaps. Why? Evolution just evolved that way. One is not stronger than the other. They've done some modeling and some tensile tests in which they figured it out. It doesn't seem to have a dramatic difference in functionality. The mitral valve is more likely to go bad on you, but not because of its structure, because of its pressure. We'll talk about that in just a second. So this is the mitral valve. So as pressure builds up here, it's going to go through the mitral valve or the left atrioventricular valve. And then it's going to be in here. Same stuff in here, a little bit different orientation, but trabeculae carneae and papillary muscles and chordae tendineae. And when this contracts, what's going to happen is I'm going to push it out of the ventricle. I'm going to push it out through this valve, which is the hardest one to see. This one is smashed back behind the pulmonary trunk. And you can't even, it doesn't even really look like it's pumping blood into the aorta, which is why you should stick your finger in there and convince yourself that the blood actually goes in there. I know it seems silly to do stuff like that, but remember that the more senses you can involve in understanding, the better recall you have. So stick your finger in there, and you should see it wiggling right about there. So this valve right here is called the aortic semilunar valve, and it leads into the biggest, highest pressure artery in the body, which is the aorta. And the aorta, of course, is going to deliver oxygenated blood to where? It's everywhere but the lungs. Okay, that makes sense? It actually even delivers a little bit of blood to the outer portions of the lungs that aren't doing gas exchange. So that makes sense? So then the blood is, of course, going to go into systemic circulation from there. So that's the basics that we've got there. Trabeculae carneae, we did all that. Okay, so now let's talk about valve function. Okay, so valves function differently than people assume that they function. And it makes sense that you would assume that they function. People think that valves open and close due to muscular contraction. They don't really. They open and close due to pressure differences on the sides of the valves. Because really they're saloon doors, right? And all you have to do is push on the saloon door. You don't have to pull on it, right, to get it to move. So what I want to do is I want to start by delivering blood back to the atria and see how we can predict when the atrioventricular valves will open, then when they will close, then when the semilunar valves will open and when they will close. And you do this in a lot of detail in phys. But it's right at the end of the semester and people are so damn rushed that they have a hard time getting it. So, venous return is constantly sort of passively delivering blood back to the atria. So remember that blood is going to trickle from capillary beds, but it's going to trickle constantly from capillary beds. So I've always got blood just sort of passively coming into the atria. Does that make sense? And as the blood starts to pool, especially if you're standing up, but it doesn't have to be standing up. As the blood starts to pool here, it's going to put pressure on these closed saloon doors, right? Okay? And as it puts pressure on those closed saloon doors, it will cause them to open. A lot of people, most people, think that the AV valves open due to atrial contraction. That's actually not true. Atrial contraction is not what causes the valves to open. As a matter of fact, atrial contraction isn't absolutely necessary for about 80% of your blood flow. You're going to feel it if they don't. So what's going to happen is these are open, and they're open because of the venous return. So what does atrial contraction do? What atrial contraction does is at the end, after the valves are already open, the atria contract and squish the last 20% of blood from the atria to the ventricles. That is why I am alive, because I have atrial fibrillation. And atrial fibrillation is just like, it's not a super major, I mean, it can kill you, but not most of the time. So what should be happening with a healthy heart is you should have atrial contraction, pause, ventricular contraction. Atrial contraction, pause, ventricular contraction, right? Okay. But what atrial fibrillation does is, imagine that I'm coordinated enough to keep doing this, and I'm not. Okay. So this is what my ventricles are doing. They're fine. They're keeping going. And then my atria, every once in a while, will do this. Just spaz out, right? And what happens at that point is my blood still going from my atria to my ventricles? It drops by about 20%, but I still got 80% of the blood still going. So I don't die, right? If it was atrial contraction that caused the valves to open, I would already be dead, right? But what does happen is I get super lightheaded and sometimes I pass out flat. And one time, one of my students had to drive me home. That was really nice. So it's like stress-induced, and it's because I had some infectious diseases when I was a kid. So atrial contraction occurs after the valves are already open. So now the blood's in the ventricles, okay? The function of the valves, by the way, guys, is to ensure unidirectional flow of blood through a circuit. And if the blood isn't flowing unidirectionally, then it's not going to have efficiency as far as gas exchange goes. Does that make sense to you? So the valves are basically supposed to make sure that you don't go backwards. So what's going to happen, and now this valve is open and the blood is in here. After I've squished the last 20% in, then what's going to happen is the walls are going to start to contract. And as the walls start to contract, I'm going to push on the back side of these saloon doors. And I'm going to push them. But I don't push them open the other way because of our friends here, right? And our friends here are the papillary muscles, and papillary muscles hold on. Papillary muscles actually start to contract before the walls of the ventricles do. They're like playing Red Rover. So right before the ventricles contract, they say, Red Rover, the blood's coming over, and they go like this, and they hold on. So the papillary muscles are holding on, and then what happens is the chordae tendineae and the papillary muscles, I'm going to push on the back side, and assuming those are working well, they won't flap backwards. Does that make sense? Okay, so why do those valves close? They close indirectly because of ventricular contraction, but really it's because you're pushing the blood against the backside of them, right? You're generating the pressure with the ventricular contraction, but the ventricles are not really opening and closing the valves. The ventricles are changing the pressure in the blood, which opens and closes the valves, right? So now I've closed these. I generate the pressure just a little bit higher, and then I push on the back of these saloon doors, right? And when I push on the bottom of those saloon doors, what do? They open. Exactly, right? There is a lag time between this one closing and that one opening in a healthy person, right? This one closes at lower pressure than that one opens. That makes sense? And that promotes unidirectional flow of blood. So now my blood is being pushed through the semilunar valves into the arteries, and as it's pushed into this from the semilunar valves into the arteries, I want to talk about why those things will close. Okay? So now what's going to happen after I push it into the arteries is the ventricles are going to start to contract. Anybody want to hazard a guess as to why those semilunar valves will close? What?
Was the other ones opening?
No, it's not the other valves. Okay, so I want to know, we know why these open, right?
Yeah.
Because they generate pressure on the side of them. I want to see if you guys can figure out why they close. Okay, you push blood out and you relax the ventricles. Why are these going to close?
Backflow.
Yeah, the blood tries to go backwards as this gets to be lower pressure than that. Yeah? So why don't these need papillary muscles and chordae tendineae? Because most of the blood's gone, right? It's not super high pressure. So what happens as the blood flows backwards is it gets caught in the cusps, right? But most of it's gone, right? You catch some in the cusp and it closes it, right? That makes sense? So that's sort of the functionality of the valves that you're dealing with there. Okay, so as far as heart sounds go, let's briefly talk about heart sounds. So heart sounds, what you're hearing when you hear the lubbed up of heart sounds is you are not hearing the heart contracting. You can't hear that. And you're not hearing valves opening. You generally can't hear that. What you can hear, however, is when the valves close and you slam the blood against the backside of a closing valve, you can hear that. So when you hear a heart sound, which is lubbed up, the first sound is lower and the second sound is higher and more staccato. It's sharper. So what you're hearing is the lub is closing the AV valves and slamming the blood against it, right? And then there's a pause. And then the dup is closing the semilunar valves and slamming the blood against it. Does that make sense? That's what you're actually hearing. You're not actually hearing things moving. You're actually hearing closing of valves and blood slamming against it. Is that okay? Okay, so with that in mind, I want to talk about what happens when your valves are incompetent or do not behave in the way that they're supposed to, which definitely happens. Okay, so with incompetent valves, there's a bunch of different valve disorders. I'm only going to talk about a couple of them. A prolapsed valve is one that swings too far. So let's talk about, of these pairs of valves, AV valves and semilunar valves, which of the pairs, AV or semilunar, do you think is more likely to swing backwards? Any idea?
Backwards in the opposite direction.
As in, go past the close point so that it's open the other direction. It looks like it might be semilunar, but if I tell you to think about pressure, it's the AV valves collectively because they're so much higher pressure. Why are they so much higher pressure? Well, because I'm pressing a closed chamber, right? As soon as I close the AV valves, the entire chamber is closed and I'm increasing the pressure on a closed chamber, especially in the left side where I'm generating boatloads of pressure, right? So those are really high pressure compared to the semilunar valves, in which as long as nothing is constricting my aorta or my semilunar or my pulmonary trunk, the blood is gone, right? They're not super high pressure. So as far as the likelihood of prolapse, the most likely pair of valves is the AV valves, and the most likely of the two AV valves is which one? Yeah, the bicuspid. But it's not because of the weakness of the structure of the bicuspid, it's because of the pressure the bicuspid is dealing with, right? So if a valve swings too far, it's called a prolapsed valve. There was some really famous clinical situations in which use of a certain medication caused mitral valve prolapse. Did you guys hear about some of you guys were too young to hear about it, but you can still read about it. But 15, 16 years ago, there was the huge multi-million-dollar lawsuit against Fen-Phen. So Fen-Phen is a weight loss drug. It's actually two weight loss drugs. And they were both evaluated by the FDA, phentermine and fenfluramine. And then somebody figured out that they're way more effective if you use them together. But that wasn't evaluated by the FDA. The combination of the drugs was not evaluated by the FDA, and it was prescribed relatively commonly. What apparently happened is that the combination of the drugs caused scarring of the mitral valve and can cause mitral valve prolapse. And I know this personally because my father-in-law took fen-phen and had mitral valve prolapse. His mitral valve prolapse was so bad that his heartstrings popped. And he ended up having to get a mechanical valve replacement for his mitral valve. It's right here. Some people got pig valve replacements, and when you sit next to him, you can hear the click, 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 click. It drives me bonkers. It drives me bonkers. I'm sure it drives him bonkers because he can't freaking get away from his heart. But I don't sit next to him at dinner. So that's mitral valve prolapse. It can happen with other valves. Of course, it's a big deal if the entire thing prolapses because it's going to compromise the functionality of the blood flow. Now, there's a more minor problem that's also more common, and that is a murmur. So when I was a kid, I had, before they vaccinated for certain types of things like rheumatic fever and scarlet fever, and they don't vaccinate against scarlet fever right now, I got those when I was a kid. And rheumatic fever impacts the heart valves. It likes to attack the heart valves. And when it does, it can cause scar tissue formation. And scar tissue has less plasticity than the actual tissue. I always like to think about the seals of your car when you get a brand new car, unless it's a Subaru, which are so noisy. If you get a brand new Toyota, it's really quiet, but then go through 5 years of like hot summers and salt, and then it gets really louder in there. The same thing happens with heart valves. If they get scarred, what happens is they don't seal as well. And then what happens is you can hear a little bit of blood going back through it, and that's called a murmur. And if a murmur is a lot of blood, then sometimes they'll do some interventions, but if it's just a minor amount, they'll just say you have a murmur, you need to watch that. So I have a relatively minor murmur. Murmurs, you can hear them. I'm really bad at hearing them. I can finally hear mine. But and they sound different if they're AV or semilunar murmurs. So if I listened to your heart and I heard lub, slush, dup, is it an AV murmur or a semilunar murmur? It's an AV murmur, right? Because you can hear it going backwards through that closed lub, slush, dup, right? If it's a semilunar murmur, which are less common, it's lub, dup, slush. I have an AV murmur, and my cardiologist constantly will he makes me listen to his heart and listen to my heart. And finally I can hear the damn murmur. And now he's like, if you listen closely, you can tell whether it's left side or right side. And I literally cannot hear it. Eventually I just nod. I'm just like, fine, yeah, totally get it. I don't know what the hell you're talking about, but I cannot hear it. He's like, listen to this side, listen to that side. I'm like, whatever. I'm just trusting. So that's a murmur. Stenosis is a narrowing of valve that can be due to scarring or infection. Bloodborne pathogens like to attack your heart. I told you that cardiovascular health can be predicted by oral health. And then there's also certain predictable communicable diseases that like to attack your heart. And that's one of them that I got when I was a kid. If you have a severe murmur, it's really hard on your cardiovascular system because you have to pump more frequently and harder to overcome. Does that make sense? Okay, so now let's just talk briefly about the great vessels of the heart, and then we'll talk about blood supply of the heart. So what do people mean when they say great vessels of the heart? The vessels that are directly attached to the chambers are considered the great vessels. So the great vessels of the right heart are the superior and inferior vena cava and the pulmonary trunk. Yeah? And the great vessels of the left heart are the left and right pulmonary veins and the aorta, right? So the aorta is right there. The aorta, of course, doesn't look like it's attached to the left heart, but it actually is attached to the left heart. Now, the coronary sinus, which we're about to talk about, people argue about whether that's a great vessel or not. I don't care whether you categorize it as a great vessel or not. Some people say it's not a great vessel because it's only delivering blood back from a single organ. But other people say that it is because it's delivering it directly to a chamber. And I think it's semantics. I don't really care. But we will talk about its functionality, and its functionality does matter. Okay, so here's something that I was confused about for an embarrassingly long time. I don't know why. the limits of my intelligence or some kind of assumption that I had. I couldn't figure out for an embarrassingly long time how the hell anybody could ever have a heart attack due to lack of blood flow to the heart when the damn thing is full of blood all the time. Like, I don't understand what you're talking about. Are you telling me like for 2 minutes it didn't have any blood in it? Because I had just a miscomprehension. I did not comprehend the fact that the blood that is going through the chambers of the heart does not actually deliver blood to the capillaries of the heart. So the blood in the chambers is for other people, right? It's delivering it elsewhere, okay? So the blood that goes to the heart actually doesn't come directly from the inside of the heart, it comes from the outside of the heart in, and we'll talk about that in just a second. First, let's talk about how greedy the heart is. The heart takes a disproportionate amount of blood flow. So it accounts for around 1/200th of your body weight, but it eats up about 1/20th of your blood flow. The only thing that's as greedy is your brain at rest, and then your skeletal muscles are super greedy, but only when you're exercising. They're really variable in their blood flow needs. Okay, so how do you get blood to the heart, and why don't people understand this intuitively? The reason that you don't understand it intuitively is because you can't see how it's hooked up from looking at the heart. So what you have to do is ghost it to see the blood flow to and from the heart. So what we are going to do, conceptually, you guys told me that the heart should get what kind of blood, systemic or pulmonary? Yeah, it should get systemic blood. As a matter of fact, the heart is its favorite organ, so it gives itself first dibs. So when you pump blood into the aorta, the very first 2 branches off of the aorta are the left and right coronary arteries. And they deliver blood to the heart muscle capillaries from the outside in. Does that make sense to you guys? Okay, so they are going to deliver blood to the capillaries. So, but you can't see those. So what I want you to see is this one right here runs behind the pulmonary trunk and under the left auricle. You can't see where it's coming from, right? It's even a pretty hard dissection to do. You kind of end up damaging the entire heart by pulling out this dissection. And then this one you can't see where it's coming from because it runs under the right auricle. So you can't really like see that this is where the blood flow is coming from. Otherwise people might be able to intuitively understand this. So what's happening is the blood is going to be delivered to the ventricles. There's also a couple of branches to the atria, but they're really tiny, so you don't learn their names. So you're going to deliver blood to the ventricles from the outside of the heart in. The first squirt of systemic arterial blood goes to the heart itself. Okay, so this is the left coronary artery, and so let's learn the vessels of the the vessels that deliver blood to the left heart. Okay, so this one is going to be off of the ascending aorta, these first 2 branches. This one runs behind the pulmonary trunk, under the left auricle, and then the only place that you can actually identify the left coronary artery in an intact heart or a model is right there before it branches. You can't see the rest of it, okay? So right there before it branches, it's called the left coronary artery. And then after it branches into this vessel, this big honker right there. And that's called the anterior interventricular artery, or clinically they usually refer to it as the LAD, the left anterior descending artery. Okay, this one is a really, really important artery because it delivers the vast majority of blood to the left ventricle. Right? That's why when that sucker gets blocked, they call it the widowmaker. Okay, because it'll kill you dead. Okay, so that is the anterior interventricular artery, or the LAD, the left anterior descending artery. And it sits in a sulcus that you already learned. What was it? Anterior interventricular sulcus. And then the other branch right here, this circumflex artery, is going to run in the coronary sinus around the left side to the back. That's the circumflex artery. Good? Okay, so there are other arteries, but they vary a lot between people, so the only ones that you guys are going to learn are the ones that are relatively consistent between people. Okay, so this guy right here goes under the right auricle and then runs all the way around to the backside in the coronary sulcus. And this entire thing right here, all the way around until it branches down, is called the right coronary artery. It's called the right coronary artery almost the whole time. And it gives rise to 2 branches that you need. This branch right here is called the marginal artery, or sometimes the right marginal artery. I don't know, eventually they figured out that you didn't have to call it the right marginal because there wasn't a left marginal. So sometimes they leave off right and sometimes they put it on, and I don't really care which one you do. It's the only marginal artery that you have. And it's like almost at the margin of the heart. Okay? And then this other guy right back here, do you see that? That is called the posterior interventricular artery, and it runs in a sulcus that you already learned, which was posterior interventricular sulcus. So it doesn't look like this when you look at a real heart. When you look at a real heart, first off, there's so much fat on the surface of a real heart that you have to really dissect it off before you'll see the vessels at all. And then the vessels, you're going to have to really look at them. And in order to tell an artery from a vein, you're going to have to get your fingers involved to figure out which one's which, right? But you can see them here. So this is the anterior interventricular artery. This is the anterior interventricular artery. What's this one right here? Circumflex. What is this one all the way around? Right coronary artery, this guy right here, marginal or right marginal. And then the others, you have to flip it around the backside to see. If you flip around the backside, you don't have to learn this one. This guy right here is what? Artery? Posterior interventricular artery. And then this guy is still right coronary. Right coronary as it wraps all the way around. That makes sense? Okay, so what we just did with these arteries is we delivered systemic arterial high oxygen blood to the myocardial capillaries. I mean, it's going to branch into arterioles and stuff, but eventually it's going to get to the myocardial capillaries from the outside in. We're going to go deep into the super vascular myocardium, right? And then we're going to do gas exchange as we go through, right? And we're going to gobble up all the oxygen and generate a whole bunch of carbon dioxide. And then we have to get that blood back to what chamber of the heart? Right atrium. Okay, so now with these vessels, I want you to think about which direction blood is flowing. So these are the cardiac veins. By the way, most common mistake when people are identifying this stuff, the arteries generally are coronary and the veins are usually cardiac. Coronary refers to a crown, and somebody thought this looked like a crown all the way around the heart. Corona, crown, okay? So don't use those terms interchangeably because you'll actually get it wrong. Some of them are called coronary and some of them are called cardiac. Okay, so this guy right here is called the great cardiac vein. And it is delivering blood back from the left ventricle. Which direction is blood going to be flowing through that one? What do you think? Think about where it's going to end up. Great cardiac vein. It's going up. Yes, it's going up, right? So it's going to go up back toward the coronary sinus. It's called coronary sinus all the way around the back, just so you know that entire blood vessel that's running in the coronary sulcus is called the coronary sinus. So it's delivering it all the way up into the coronary sinus. And if you start at the front and then work around the back and then back to the front, you won't have to memorize which one's great, middle, and small. I'll get you in just a second. And then this guy right here, on the back side, it's ghosted, it's on the back side. That's the middle cardiac vein. And then keep going around, and this one over here is the small. So if you start from the left side and go around the back to the front side, then you go great, middle, small. What was your question, Lincoln?
Didn't you just say that coronary is a reference to an artery? But this is a coronary.
No, I said these arteries, somebody thought all of them looked like a crown, so they called those coronary.
Oh, you're saying they each have their own specific name.
All of the arteries are called coronary arteries, and most of the veins are called cardiac veins. This is the coronary sinus because somebody thought that the entire coronary sinus looked like a crown too.
But
So it's not.
cardiac isn't specific to.
No, it's not. But
vein
most of the veins are cardiac, and most of the arteries are coronary. So you really just have to kind of get used to it. The three veins that are called veins on the surface are all cardiac. So it's great cardiac, then middle cardiac, then small cardiac. Does that make sense? And then the entire thing dumps into the coronary sinus, and then the coronary sinus dumps into the right atrium, right there. Okay, so just one more time, let's find it on an intact heart because it doesn't look like that and it's not ghosted. Okay, so here's an intact heart. You know what this artery is called? Anterior interventricular artery, or the LAD. And you know what the sulcus is called? What is this vein? The great cardiac veins runs with the anterior interventricular artery, right? That makes sense? And then it's going to dump into, you can't see it right here, but it's going to dump into the coronary sinus, right? Okay, and then this vein you can hardly even see. You'd have to lift up the atrium to see it a little bit. But what is that one? The small right here dumps into the coronary sinus. So the coronary sinus runs all the way around, right? It's what everything dumps into. Now, the small doesn't usually run right with the marginal, but it usually runs near the marginal, right? So here's the marginal artery, and here's the small cardiac vein, right? And then if you flip it around the backside, you'll see the rest of them. Flip it around the backside. This guy right here is running with the posterior interventricular artery in the posterior interventricular sulcus. What is that one? That's the middle, yeah? That's the middle, and it dumps into the coronary sinus, yeah? And then the coronary sinus dumps into the right atrium. Good? Okay, so this is your first blood trace that you are going to do. You're going to do other blood traces next in the next chapter, but a blood trace is when I ask you to conceptually list out the vessels I would get to. For instance, I could say start from the aorta or start from the left ventricle. So just listen, you don't have to do this one specifically, and this is not the last trace you're going to do. But if I asked you to start from the left ventricle and list the valves and vessels you would go through in order to get back to the right atrium, right? So let's just do that one out loud. You ready? Okay. So, to get to blood to the anterior wall of the left ventricle and then back to the right atrium. Okay. So one more time, let's say it a little more clearly. What I want to do is I want a trace that takes me from the left ventricle out to deliver blood to the anterior wall of the left ventricle, and then back to the right atrium. Do you see what we're trying to accomplish here? Okay, so the first thing I would do is I have to name the valves and vessels, right? So the first valve is what? The aortic semilunar valve. And then what I'm going to do from the aortic semilunar valve is to go into what vessel? The aorta, and it's actually the ascending aorta, okay? And then from the ascending aorta, where do I go? I'm trying to get blood to the anterior wall of the left ventricle. Where do I go? Get me there. Left coronary artery, and then what? Anterior wall of the left ventricle. Anterior interventricular artery. You guys with me? Okay. Deliver it to the capillaries. Then the last part of your trace was to deliver it back to the right atrium. So now let me get it back. Okay, went through the capillaries. Capillaries are invisible. They're not on the picture, but they're an important part of the story, right? And then now what vessel do I go into next? Great cardiac vein, and then where? Coronary sinus, and then right atrium. That makes sense? That's a blood trace. Blood traces are just you conceptually going through them, and you will have like 6 different blood traces that you are going to do in not the heart lab, but the next lab after that. So this is like the first time that you're thinking of traces. A lot of times they'll make you go through the left arm and then all the way back, or through the liver or something and all the way back. And we'll work on this in lecture too. Does that conceptually make sense to you? Okay. I love blood traces. I don't know why. They're just like making a boring picture into a story, and I like stories. Okay, so let me tell you something interesting that I also think is worth thinking about. Okay, so your heart at rest, let's say your heart is beating 70 times per minute at rest, and that's just an average. No matter how quickly your heart is beating, it spends about as much time relaxed as it does contracted. So about 50-50. So if you increase it, you're not going to decrease the percentage of time that you spend relaxed. So contracted and relaxed. And that is because heart relaxation is just as important as heart contraction. Of course, we know what's going on during, for instance, ventricular contraction as you're pushing blood out to systemic organs, right? Yeah? So what is important that's going on during, for instance, ventricular relaxation? What's going on? Filling, and that's super important, right? Because you don't have a damn thing to pump if you don't like get anything in there. So ventricular filling, but then there's more important thing that is going on during ventricular relaxation, and that is this. So when the ventricles contract, you push blood into the aorta and then into the left and right coronary arteries and the arteries that branch from them. You guys with me? So you push it in those with ventricular contraction. But what happens is at the same time as you're pushing blood into those arteries, you are compressing the capillaries. because you are using the pump to the pump. Okay, so you're pushing it into the arteries, but you're not actually delivering it to the capillaries until you relax the heart, right? It's like squishing a sponge underwater. When do you actually get the water into the sponge? When you relax your hand. So what happens is if somebody gets into a ventricular fibrillation situation, right? Why the hell do you have a heart attack if you're just doing this? Well, the combination of the two things that you just said. If you're doing this and you're not relaxing, then you're not delivering much blood because you're not filling. But in addition, you're also compressing the capillaries of the heart and cutting off blood flow to the heart itself. Does that make sense? So ventricular relaxation is just as important as ventricular contraction, right? And ventricular fibrillation will kill you quickly. How quickly does it kill you? Yeah, I'm asking you.
Oh, I don't know. I'm sure it's different for everyone, but I was going to ask you a question. So you know that only 2 shockable rhythms are VFib and V-attack? Like I always wondered why it's just that.
They're not the only 2 shockable. They're the only 2 like really easily shockable. I got into atrial fibrillation for 16 hours one time and they were about to shock. Those 2 are going to kill you. So what they do when they shock is they stop the electrical activity of the heart and wait for the SA node to pick it up. So it better be pretty damn bad for them to be willing to stop your heart.
Atrial, you have to pace it more specifically. With ventricular, you don't have to pace it in the same way. Does that make sense?
What do you mean pace it?
So like because if you shock the heart when it's in a specific orientation, it doesn't. It can kill you. Like the shock itself can kill you depending on the orientation of the heart or where it's at in the beating process. But if it's, and I think that has more to do with the fact that it's coming from the atria, or if it's an atria.
Interesting. I don't know the answer to that question. That one's a clinical one that's like outside of my depth. I do know with atrial fibrillation that they will not consider it very soon. But I was hospitalized for 18 hours and I was in atrial fibrillation and they were about to shock me for it. But with ventricular fibrillation and ventricular tachycardia, you're about to die anyway, right? You're like literally about to die. They might as well like shock you because they've got to stop clearing. Yeah, is it minutes? I was wondering how quickly it was. I don't know. Okay, so last thing on this stuff is just to talk about innervation and electrical activity of the heart, right? So the heart is a mechanical organ, but the heart is also an electrical organ. So we have the mechanics kind of down, right? You got atrial contraction, you got the opening of the valves and the closing of the valves and ventricular contraction. That's a mechanical activity of the heart. The electrical activity of the heart is actually what causes the mechanical activity of the heart, in part, though. So first thing is to tell you a couple of things about your heart. And one of the important things is that your heart will actually contract in the absence of nervous system stimulation because your heart actually has its own internal capacity to produce electrical stimulations because it has cells in it that are called pacemaker or conducting cells, and these cells spontaneously generate an electrical stimulus and then send it through gap junctions to their friends. And what happens is the electrical stimulus, which is called an action potential, is what causes contraction, right? So you send a signal and then it causes contraction. That makes sense to you? So your heart, I think one of the students said last time, I guess she's in the other class, your heart will contract in the absence of nervous system stimulation. It's not like skeletal muscle. Skeletal muscle cells won't do a damn thing unless you tell them every single time specifically what to do. Cardiac muscle cells will. That's why, for instance, you can have a heart transplant and not hook it back up to the nervous system, and then assuming that it was a successful heart transplant, it will still beat. You can't control its rate much. It will beat about 100 times per minute because what happens is 100 times per minute, this blob of pacemaker cells, yes, that's the anatomical term for it, blob of pacemaker cells, right there is called the sinoatrial node. 100 times per minute, it spontaneously starts an electrical stimulus and then sends it through gap junctions to its neighbors, right? What this is, even though this looks like nervous tissue, it's not nervous tissue. It's really specialized cardiac muscle cells that can generate spontaneous electrical stimuli. So these are called pacemaker cells. If you've got a blob of pacemaker cells, it's called a node. So this is the sinoatrial node. You find that just inferior to the opening of the superior vena cava in the right atrium. Okay? And what it does is it communicates with its neighbors. It starts an electrical stimulus and then it sends it out through its neighbors. And when it does that, the neighbors will contract in the order that they got the stimulus, which means that all things working properly, the atria should contract in what order? How should they contract? If they starts at the SA node and then does this. Top down. That's exactly what you want them to do, right? Okay, so the atria will contract top down. And about the time that the stimulus gets to the bottom of the atria, this is another node called the atrioventricular node. It's in the interventricular septum near the bottom. That one was on its way to an electrical stimulus anyway, but the SA node is the boss in a healthy person. And so that one was like starting its electrical stimulus, and then the SA node says, do it now. It's like your kid was on his way out the door taking the garbage out. And then you say, take the garbage out, and then they lose all credit, right? Oh, I was doing it spontaneously. The AV node was always about to do it spontaneously, but the SA node is bossy, and it says, do it now. So the AV node now will generate a spontaneous electrical stimulus, but this is really important. The way that the AV node works is it doesn't talk to any contractile cells. What it does is it talks to its pacemaker neighbors, and its pacemaker neighbors are in a chain. So here's the AV node, okay? Here is the AV bundle. This is just a chain of those cells instead of a blob of those cells, okay? And then the AV bundle splits into another chain. This one is the left bundle branch, and this one is the right bundle branch. Still, all pacemaker cells just in a chain, and those will ultimately talk to these fibers, which are called Purkinje fibers. So, the important thing to understand is only the Purkinje fibers will give the stimulus from the pacemaker cells to the contractile cells. That means that what happens is the SA node says atriocontractile cells contract and tell the AV node what to do. Right? And then the AV node says, I'm just going to pass this on like a game of telephone to my other pacemaker cell friends. So what happens is the AV node sends it down the middle of the interventricular septum and then up the left and right bundle branches, and then it sends it to the Purkinje fibers. This is important for two reasons. First of all, it forces a lag time between atrial contraction and ventricular contraction, which is important for filling. And then second, if it works this way, that means that the ventricles will contract in what order? Bottom and sides up, right? If you don't start from the middle, you don't start until you get to the Purkinje fibers, right? Which are down here at the bottom and up the sides. That means that once you start controlling the contractile cells, you contract bottom and sides up, which is what you want, right? Okay, so this forces a lag time of about 0.2 of a second between atrial contraction and ventricular contraction when your heart's at rest. And that's for ventricular filling. Does that make conceptual sense to you guys? Yeah? You got it? So what you end up with is you end up with a contractile process that looks like this. So the atria contract down and the ventricles contract up. Atria contract down, ventricles contract up. Does that make sense? Okay, so any questions? Okay, so the Purkinje fibers are at the bottom. So, by the way, you really cannot see any of the electrical stuff in the heart because really the cells are the same. It's just that they are functionally different. Every once in a while, like the SA node has a slightly different texture and the AV node does, but don't depend on seeing those. Primarily, you will see them in models and know that they're there. Really what they are is specialized cardiac muscle cells within the wall, so you shouldn't expect them to have a dramatically different gross anatomy. Right? And you can dissect them out if you're really, really good at it, but I've never actually gotten more than just these two in the beginning of that one before I trash the entire thing. So it's primarily a functional difference even more than a structural difference. Okay, so the last thing to talk about is, before we go any further, I want to show you guys something. I want to back up just a minute and talk about, I meant to do this right after we did the blood supply of the heart. So let's go back for just a second before we talk about innervation. So here's what we were talking about. We were talking about delivering blood to and from the heart muscle itself. Please note that this heart has tremendous energy and oxygen requirements, like tons of blood is required to the heart. Also, two other points. It has almost zero capacity for anaerobic cellular respiration, so it constantly needs oxygen. And then the other thing is that the cells are amitotic. If you cut off blood flow to them, they will die and they don't go through mitosis, which is why the damage that occurs from a heart attack is permanent damage. Your heart is permanently weakened after a heart attack. So what's going to happen here is, let's say that you're an American, and most of you are. And you have American diet and exercise patterns, which is that we tend to be less active than people from most other countries, especially as we get older. And then we tend to eat a lot of cholesterol and a lot of sugar. That combination loves to gunk up the walls of your blood vessels, especially if you add in something like smoking. And then you get nice toothpaste blood vessels like this. Okay, and you can squish them and squish the fat out of the walls of the blood vessel. So this is problematic everywhere, but it's especially problematic when you're dealing with a tissue that has tremendous blood flow needs and is amitotic, so heart and brain. So this can get so very blocked up that you can reduce a vast amount of blood flow. This is a postmortem photo of a coronary artery. The lumen of the coronary artery is supposed to be this big. Okay, I don't know what percentage blockage that is, but it looks like 90 or something. I don't know, I can't tell. And this is the plaques start out soft and they eventually become calcified and crunchy in the walls as well. So this is what we're all doing right now. So we should all probably run to your car. So what? Run to your car at McDonald's. My cardiologist insists that your cardiovascular system will forgive you anything if you wiggle a lot. You can eat anything. He's like, you can eat any damn thing you want if you exercise, right? Because exercise cleans out your blood vessels. So the combination here, by the way, is the combination of cholesterol and sugar. It seems as if sugar is really, really good at making it stick to the walls. And the bad cholesterols, which are the LDLs and the VLDLs, primarily from eating fatty animal products. In combination with low levels of the HDL, which you get from exercise and cleans out your blood vessels. So this is what actually does this. So what happens? So what happens in this situation if I have a blockage? So if I have a blockage in one of these coronary arteries, and if you have a blockage in your coronary arteries, you got it other places too, but this is the one that's more likely to kill you, that one or the one to the brain. They're tiny to begin with, and they have tremendous needs for blood flow. So let's say you got a blockage in your LAD, which is really common. So left anterior descending. So you got a Widowmaker blockage. What do you do about that? Well, first thing is that your doctor tells you long before the blockage occurred that you have high cholesterol and you should probably start exercising and eating better. And then you go, wah, 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 and you don't do any of that shit. And then later, you ask for a pill. And then 5 years, 10 years after that, you consider having angioplasty, which is when they stick a bubble in there and pop open the vessel. And then even later after that, you can, for instance, have a bypass. And what a bypass is you got such a gunked up blood vessel that it's limiting blood flow to the point that either you had a heart attack or you're at really high risk of a heart attack. And what they will do is they will take another vessel from somewhere else in your body and they will usually attach it to the aorta and then they will bypass the blockage so that your heart can still get blood. There are a bunch of different ways to do it. I'll show you a video of one of the ways that they do it. It's just a short little video, a minute long.
Your heart is a beating muscle whose main job is to supply your body's cells with oxygenated blood and nutrients. These life-sustaining supplies are delivered through coronary arteries. Coronary means crown. The artery's crown will surround your heart muscle. Coronary arteries nourish the heart, but if they are narrowed or blocked because of fatty buildup called plaque, the flow of blood and nutrients to parts of the heart can decrease or stop. When this happens, it causes the cells in that region to die and leaves the heart without the proper amounts of blood and nutrients it needs to function. Exercise and proper diet are essential in the prevention of heart disease, but sometimes surgery is necessary to revitalize the heart. Heart bypass surgery grafting is a procedure where an artery or vein taken from your leg is attached to the aorta and is used to create a new pathway to your heart, bypassing the blockage that exists in the disease section of your artery.
So they used to pretty consistently do it from the great saphenous vein in your leg, and then somebody eventually figured out that it would be much better to replace an artery with an artery than it would be to replace an artery with a vein. Although you don't have a lot of spare arteries, right? Like they're busy, right? So now even more common than this, I think now, from what my cardiologist told me, is in males, they will use the mammary artery because you're really not doing that much with it, right? You're like, I'm just going to take that and move it over. And then sometimes they actually now, instead of bypassing from the aorta, they will take like one of your intercostal arteries and just attach it to your heart. So they don't even like move it. They don't even like detach it from where it was. They're just like, oh, I'm going to put this one over here. So isn't that kind of cool that they do that now? Go ahead.
So naturally, to clean out the arteries, exercise and diet and whatnot, but what's actually happening to the arteries? Like where's the stuff going?
Oh, it cleans it out. It just gets moved through circulation and cleared by other mechanisms. It's primarily fatty, so you clear it the way that you clear other fats. Detoxify it in the liver, poop some of it out. If you can get it water soluble, you'll pee some of it out. You can clean out your blood vessels pretty well with exercise if they're not too crunchified already. For some reason, smoking, it makes it very, very difficult to clean them out. So if you are a smoker, even if you eat well and exercise, you probably have toothpaste in your veins, arteries.
Living in Utah with this lung, does it crunchify your arteries?
I don't know. That's a good question. I should ask. It's not cigarette smoke, huh?
Did you say the lungs look the same?
The lungs of people who have been raised in the valley look like smoker lungs. It's very disturbing for me to think about. I have a lot of cognitive dissonance about that. I'm like, no, don't go there. Go ahead.
Do you think it's different if you move here as an adult with fully developed lungs, or do you end up looking the same?
I don't know. I don't know the answer to that. I wonder if anybody's ever done a long-term clinical study of that. I should ask the body donor program if they were like, let's just like gather lung tissue from like as many people as we can. But the donors don't actually have to tell us very much of their history. They just provide what they want to provide. So we'd have to have some deeper information about them. Usually all we get from the donors is first name, age, occupation, and then every once in a while they'll volunteer something about their medical history. You should, by the way, go see on the cadavers because we have at least one cadaver on each campus that has a bypass. And by the way, bypass, the whole like quadruple bypass. One of my students in the last class told me that her uncle had a septuple bypass. Is that 7? That is such bad news, right? A septuple bypass. So isn't that 7, right? Okay. So 7 different bypasses, right? It just depends on how many blockages there are that you have to bypass. So go look at the cadavers. We have a cadaver with a pacemaker on Redwood campus, and we have cadavers with bypasses on both campuses, and it's pretty damn interesting. I like to try to follow them out from where they're going to see if I can figure out which artery they were bypassing. It's just to follow them out. It's kind of a good way to practice whether you understand the arteries. Okay, so last tidbit, and then I'll let you guys go, is just to talk about what the nervous system has the capacity to do to your heart rate, right? So what the nervous system has the capacity to do to your heart rate is it is like the accelerator, the gas pedal on your car. It doesn't do a damn bit of good if you haven't turned the thing on, right? So you have to turn it on, and that's the SA node's job. And then once you have turned it on, you can increase or decrease the rate, right? So your nervous system does not have the capacity to start or stop your heart. The starting or stopping of the heart is really, really limited to the SA node. Actually, you can stop it. Starting is limited to the SA node. So when they, for instance, shock you, right, they are not trying to restart your heart. They are trying to stop it from misbehaving and hoping that the SA node remembers how to behave. So the SA node really has to do it. But once the SA node is going, right, you can increase or decrease the rate. And the way that you can do that is that the nervous system is hooked up. And part of this is stuff that you know. So the motor innervation of the heart is controlled by which part of the brain controls your heart rate? You know this. Medulla oblongata, okay. And that Pons is respiratory system. Medulla oblongata controls your heart rate by primarily controlling which branch of the efferent nervous system? Autonomic, exactly. So let's put together 3 different sets of information about anatomy, right? Let's put it all together, which is why I teach this one after I teach the other 2. So let's talk about what is going on. This looks outrageously complicated, but it's really not that bad. So the deal is, in the medulla oblongata, there's some gray matter, you know that, right? and there's gray matter in the medulla oblongata. One of the chunks is called the cardioinhibitory center of the medulla oblongata, and one of them is called the cardioacceleratory center. I'll bet you can figure out that the cardioinhibitory center is attached to the what, sympathetic or parasympathetic? Parasympathetic. Okay, so here is what you are doing with your heart right now, because you guys are just chilling, right? So what's going to happen is, in order to slow down your heart from the 100 times per minute that it spontaneously generates to the 70-ish that it's probably doing right now, and if you're in really good shape, it's 60-ish. What's going to happen is that part of the medulla oblongata sends out a stimulus through the vagus nerve, right? And then the vagus nerve is going to go to the SA node and the AV node. And what it's going to do, two vagus nerves right here, the vagus nerves are going to release a neurotransmitter that slows down the electrical activity of the heart. That's what's going on right now, okay? So your heart without any nerves on it would beat about 100 times per minute, okay? So now you can understand that what's happening on a general basis, a pretty dependable basis with your heart, is that the parasympathetic branch of the autonomic nervous system is controlling what it's doing, right? But now you're going to get up and walk to your car. And if it's out in Egypt, right, you may need to bump up your heart rate just a little bit to get there, right? So what's going to happen then is I am going to decrease parasympathetic stimulation. If I decrease parasympathetic stimulation, it will slowly go up to 100. Yeah? Decrease parasympathetic stimulation. What if a tiger chases me to my car? Then I need to go at above 100. So in addition to decreasing parasympathetic stimulation, I'm going to have to start sympathetic stimulation. How do I get that message down? I start with the cardioacceleratory center in the medulla oblongata, and I send it down to the thoracic or lumbar region. It's actually T1 through T5 that controls the heart. Send it down to that region, right? And then just follow me for a second. Then I'm going to send it out to the sympathetic chain ganglia, yeah? And then I'm going to send that to the SA node and the AV node. And what that's going to do is increase my heart rate to get away from the tiger. That makes sense? So this is like all three anatomy concepts put together. This figure is not really that bad though, is it? Right? It's relatively simplistic after you like look at it for a second. Does that make sense? So that's how their sympathetic and parasympathetic nervous system innervates the heart. The last thing is to talk about whether your heart has, this is all motor innervation of the heart. Does your heart have any visceral innervation? Yes, your heart can hurt. It has visceral sensory fibers, and when your heart has, for instance, low blood flow, like angina, it can actually hurt because of this visceral sensation. And apparently heart attacks hurt too. I mean, doesn't sound like they'd be very comfortable, but I don't have any personal information about how a heart attack feels. Probably doesn't feel very good. We'll stop there for today. The last things we'll talk about next time are little bits of abnormalities of conduction, and I'll probably read a little bit about what they were talking about before next time, and maybe I can do a good job of teaching you guys. But two things. One, don't come to office hours today if you want to look over tests because I forgot them, sorry. And then two, please bring both sets of notes, the blood and vessels, because I haven't quite decided which one I want to do yet first. So bring both of them next time and then I'll decide. I want to make sure it correlates with lab really well, so I may flip them and I may not. So bring both of them next time.